of participants, and we can start the series again. So it's really great. And I would like also thank at this moment to Stella and to Pedro, because actually I'm not sure that I deserve to have the honor to chair the webinars anymore, but it's really a great honor. But I am not the chair of the action anymore. Coming back to the point, today webinar will be, today's lecture will be given by Evrim Umut, whom personally I know very well, because as you can read in his bio, he spent 3 years in my group. And he was currently in Dokuzur University in Izmir, in Turkey, Department of Medical Imaging Techniques. And the lecture will be about polysaccharides-based hydrogel microparticles, which is a kind of fancy and complicated systems, and I suppose something really new for us, because we did not have many lectures of this type of subjects. So Evrim, the floor is yours. I'm gone.
Thank you very much, Danuta. As you said, it is great that although the cost action, Aerolax cost action has ended, that we are still able to meet here, and I hope that these the webinars become a tradition in time. So I also thank you in person, to Pedro, to Gianni, to invite me to give the first lecture in this new series. My talk will be about the water dynamics in polysaccharide-based hydrogel dynamics. So this is the outline of my talk. So in the beginning, I will give as the motivation the applications of polysaccharide hydrogel microparticles. So what are the benefits of studying such systems? And then as a model system, we have studied the Inulin hydrogel microparticles. So at first, I will very briefly mention about the synthesis of Inulin hydrogel microparticles. Then, as the first step of characterization, I will speak about the structural and thermal characterization. So I will share you some images, thermogravimetric analysis and differential thermal analysis data, differential scanning calorimetry, and bed surface area analysis. Then, of course, a great majority of the study is focused on NMR. So I will present the high-resolution NMR spectroscopy results, the low-field T2 measurements, diffusometry result, and of course field cycling NMR relaxometry measurements. And at the end of my talk, so I will again briefly discuss the theoretical relaxation model that we have used for the fitting of the nmrd curves obtained by field cycling NMR measurements. And I will conclude my talk. So, the polysaccharides, as you know, are some sugar derivative biopolymers. So, and they have a high natural abundance. For example, they exist in microorganisms, seaweeds, plants, insects, and animals. And in these living bodies, they play a crucial role in realizing some cellular functions. For example, as well-known, starch works as the energy storage component in cells, or chitin or cellulose is responsible to preserve the cellular integrity of some cells. And And in addition to these, of course, polysaccharides have very unique features, promising features. For example, they are, since they are from nature, they are biocompatible, they are hydrophilic, and they show thermoresponsive gelation, thermoreversible gelation properties, and also some biological activities like antioxidant, antibacterial, anti-tumor, or antiviral functions. And due to all these functions, they are used in pharmaceutical applications, in food applications, and in cosmetics very commonly. However, they have some drawback as well. So they are easily undergo enzymatic degradation in physiological media. And they don't have natively any mechanical strength, mechanical stiffness. So for these reasons, they are natively not suitable for some biomedical applications, for example, in drug delivery or tissue engineering applications. But one can prepare hydrogels by cross-linking of these polysaccharides. And actually, this is very easy because these polysaccharides have almost all polysaccharides have hydroxyl groups, and some of them has even carboxylate and amine or amide groups. So they can be easily derivatized, and they can be easily cross-linked among each other. And these hydrogels built by the cross-linking of polysaccharides both show mechanical stiffness, and as well they can absorb water in very large amount, almost hundreds of times of the larger than their dry mass. So it is, and actually it is even better to synthesize these hydrogels, polysaccharide-based hydrogels in micrometer size or in nanometer size. So in this way, they also utilize the size advantages and enhance surface to volume ratio at this micro scale. So, and then such systems, such microgels, polysaccharide-based microgels can be used in biomedical applications, in stimuli responsive or sustained drug delivery. For example, if I should very briefly mention about the sustained drug delivery, that you can load and release a high amount of drug on these microparticles due to the enhanced surface to volume ratio. And again, due to the size advantages, they can get very close to the proximity of the diseased tissues and release their drug content in a long time interval. And this is actually very much needed, especially in cancer therapy, so that the body can tolerate the amount of dose in this large time interval. So another applications is the food application. So in food applications, such microparticles, hydrogel microparticles can be used in fat replacement in dietary products, or for the regulation of gastrointestinal functions and satiety control. Again, in these applications, it is similar to the case in biomedical applications. The reduced fat or some prebiotics can be loaded on microgels, and they can release this content in the digestive tract with some swelling and deswelling mechanisms. And as I mentioned in the beginning of my speech, as a model system, we studied Inulin, microparticles, and it is actually very suitable to form hydrogels, because as you can see here in the molecular formula, it has many hydroxyl groups, so and for this reason it is very hydrophilic, and it has a large, high natural abundance. It is found in more than 36,000 species of plants, and it is used for energy storage. So, if I should mention about the synthesis, the inulin-based microparticles were synthesized by microemulsion method. So, in this method, the AOT is used as a surfactant. So, in this case, as you can see here, the hydrophilic parts look at the center, and the hydrophobic parts are direct oriented outwards, building the water in oil micelles. And in this case, the polymerization reaction is governed inside these micro-sized micelles. So by adjusting the size of the micelles, so it is possible to also tune the size of to control the size of the particles. And of course, after the synthesis, as the first step of characterization, we look at the scanning electron microscopy images to look at the morphology and the particle size distribution. And as you can see here, we have a very nicely shaped, spherically shaped particles, and actually they are rather polydispersed. As you can see here, we have a large wide particle size distribution. We have particles as large as around 50 micrometer, and we have particles as small as 100 nanometer. So, and of course, since we are particularly interested in the water, in water swollen gels, microgels, there are two places that the water can be located. So the first is, of course, the interconnected network of pores inside the particles, and actually the swelling of the gels occurs by the penetration of water, filling of the water to these pores. And the second possible place is the interfacial surfaces, the volume enclosed by the interfacial surfaces of the particles. So, because as you can see here, we have some agglomerates, and particles are touching to each other, and in this case, water can be also entrapped in these volumes. And if you consider actually the polydispersity of the particles, these places also show a multidimensional scale. So, to investigate the pore size distribution in microgels, we performed bed Braunauer Emmett-Teller surface area measurements. For the people who doesn't know much about these measurements, so first, prior to the experiment, a thermal treatment called degassing is performed. So in this case, one keeps the sample at temperature 75 Celsius at 6 hours. So in this interval, you allow that the moisture, the water vapor that is absorbed on the pore surfaces and also on the particle surfaces is evaporated. And then after the degassing in the experiment, so in liquid nitrogen bath, a nitrogen gas is given, and by changing the nitrogen gas pressure, one measures the absorption and desorption amount on these surfaces. And actually, by using the suitable theory, one can obtain the pore size distributions either from the absorption or desorption curves. And here, if you look, for example, that we have take reference the absorption curve, and as I told you, there are several models, theoretical and models to calculate the pore size distribution. And by calculating Barrett, Joyner, and Halenda model, we obtain such a pore size distribution. As you can see, we have a multimodal distribution that one population is around 7 nanometers, and we have also smaller pores as well in our microparticles. And another step, we also look at the thermal behavior of our gels. This is important because when we are doing NMR experiments in a temperature range, it is generally useful to know the limits that your sample doesn't change its structure. And in our case, for example, in differential scanning calorimetry measurement, we see that in the first heating curve, we see an endothermic peak that is located at around 85 Celsius. And this is the indication of a glass transition of these gels, so which is telling us that one should not get close at this temperature when doing NMR experiment. And in another experiment, we performed thermogravimetric analysis and differential thermal analysis experiments. And as you can see here, we are seeing 70% of weight loss until the 100 Celsius. So this is due to the evaporation of water that is inside the gels. So it is again telling us something, that if we want to investigate the water behavior in these gels, we should not go so high temperatures, because then the situation of the water localization can be changed. I mean, from fully saturated pores, by the evaporation at high temperature, we might reach to partially saturated, partially filled pore situation, which will affect the behavior indeed. And in this TGA data, we also see another weight loss located at 250 degrees, and this 10% of weight loss can be associated with the degradation of the inulin gels, inulin matrix itself. And of course, we performed NMR measurements. As the first step, we collected the high resolution NMR spectra. So here you see the proton NMR spectra collected at 300 megahertz. So here the x-axis, the frequency axis, is not calibrated, but one can say that we are seeing the water peak, and actually this also shows that the dominant signal is coming from the water inside the particles, and we do not see any signal from the inulin matrix itself. But we see another thing, that we see that there is some splitting in the water resonance peak, and actually the chemical shifts in a few ppms is obviously due to the presence of different fractions of water. So, for example, for the water that is close to the pore walls or the particle surfaces, these water molecules are feeling another local field that is induced by this matrix, and this gives rise to this chemical shift. And as a second test, we also measured T2 at low field. So here you see the measurement that is performed at 13.6 megahertz at room temperature that we obtained by using CPMG sequence. And we see that the T2 of the water molecules is 48 milliseconds, which is actually much shorter than the value for bulk water, because as you know that for bulk water, T1 and T2 are very close to each other, which is around 2.5 and 3 seconds. And here we have a T2 of around 50 milliseconds. And this can be explained simply with the two-phase phase exchange model. That this is the model used in many porous systems. Here, as you can see, that the measured T2 is actually the sum of the bulk water T2 plus this contribution, which accounts the T2 of the surface and also the geometrical parameters of the surface. So that, for example, if we have a water adsorbed water layer on the surface of the pore with some thickness of lambda, and the S is the surface area of the pore, and the V is the volume of the pore. The measured T2 is given as here. So we are measuring a mixture of these two effects. And so due to the confinement, our T2 is much shorter than the bulk value. So, and then, of course, to understand the diffusivity of the waters, we performed diffusometry measurements. So, in these pulse field gradient NMR diffusometry measurements, we used stimulated echo sequence. As you can see here, this is a sequence constituted of 3 90-degree pulse, and between the two 90-degree pulse, after the first excitation pulse, one applies a gradient with a duration of a little delta. And this gradient introduced a dephasing to the spins. Then after the second pulse, it is waited for an interval of big delta, and after the reading pulse, an equivalent gradient pulse is applied, and the echo is recorded. So during this sequence, actually, the spins that are not stationary in the sample does not contribute to the echo signal. So a decay of echo, in addition to the T2 relaxation, is recorded. So by changing the gradient strength and reading the echo intensity for each gradient strength, one can calculate the diffusion coefficients by using the well-known statistical-tenor equation, as you can see here. Here, one should also say that the diffusion of the confined water is not like the bulk water diffusion. So it is some sort of restricted diffusion. So for this reason, here especially this waiting time, big delta is important. And for this reason, to have an accurate value of the diffusion coefficients, we also repeated the measurements for 3 different values of big delta. So these are the results of the diffusometry measurements. As you can see, it is repeated it is performed at 3 different temperatures. And the first thing that we see that the diffusometry data cannot be fitted with mono-exponential or b-exponential functions. So that we were able to fit the data with 3 different contributions characterized by D1, D2, and D3 self-diffusion coefficients, and where each of them is weighted with alpha 1, alpha 2, and alpha 4 weighting coefficients, which are corresponding to proton populations of each the different water fraction. And actually for the lowest temperature, we performed an independent fit and obtained the alpha values, and for the rest of the temperatures, we assume that these proton populations does not change, and with this assumption, we fit the data in other temperatures. And this is the results. That we see that the first diffusion coefficients, D1, the first process, has the diffusion coefficient very similar to the bulk water. And the second diffusion process is on the scale of 10 to the minus 10 meters square per second. That is 1 order of magnitude smaller than the bulk water. And actually we can associate this diffusion with the, if we remember our sketch, we can associate this second diffusion process with the water diffusion, with the water that is diffusing on the surface of the particles. And the third diffusion process is the 2 orders of magnitude smaller than the bulk water, and this we can associate with the water that is diffusing inside the pores of the particles, because this water is feeling a much higher degree of confinement and a random orientation of pores. So it is also physically sound that this can be associated with the water in the pores. So, and by drawing an Arrhenius plot, we also calculated the activation energies, as you can see here. And if we look at the populations, actually, we also see that the bulk-like water occupies the greatest amount of water inside our system, which is actually logical. And the second comes the water molecules on the particle surfaces, and as third, the water that is filling inside the pores. And of course, we have performed field cycling relaxometry measurements. I think most of the participants very well known about this FFC NMR relaxometry measurements, but for those who are not familiar with that, FFC NMR measurements allows us to measure the relaxation times at very low magnetic fields down to the field or down to the Earth's magnetic field. So this is possible by polarizing your sample under a repolarization field, and then once switch the magnetic fields to a lower value that you left your recipient system for the relaxation, and then you switch back the fields at a fixed value where you apply your RF pulse and read the FID values. So, and by changing the tau delays and the value of the relaxation field, it is possible to obtain plots of spin lattice relaxation time as a function of the magnetic field or Larmor frequency. For stellar machines, it is electromagnet, so there is no permanent magnet. So, and it is also possible to use a non-polarized sequence that you for relatively high frequencies, high magnetic fields where your NMR signal is reasonably high. You don't apply any prepolarization fields. You just switch from the relaxation field to the acquisition field, and you obtain your FID. So in our measurements, the switching time was typically 3 milliseconds. The polarization field in terms of proton Larmor frequency was 25 megahertz, and the acquisition field, which is fixed, that the sample is tuned and matched, is 15.8 microseconds, and the pulse 90-degree pulse length was 10 microseconds. Well, so here you see the nmrd curves that is obtained on water swollen Inulin microgels. So there are two things that is important. The first is if we compare this with the NMRD profiles for the pure water. In pure water, as you can see, the profiles are rather flat, and the relaxation rate is small due to the very fast motion of the molecules. So it's just if you put this values here, it will be just coincide to the bottom of the NMR of the sketch here. So the first observation that we have a high dispersion, and at low fields we have spin lattice relaxation rate that is almost 1 order of or 2 orders of magnitude higher than the pure water. This is obviously due to the fact that the water is confined in our sample. And the second thing which is more interesting is that if we look at temperatures, that with increasing temperature, actually the relaxation rate is also increasing. And actually this is opposite to the general case that with increasing temperature, the molecular dynamics gets faster and the relaxation rate gets smaller. So this inverse temperature dependence here can be explained by the chemical exchange, water chemical exchange that is dominating the water relaxation process. I mean, with increasing temperature, the chemical exchange of water with the water that is absorbed on the pore surfaces or particle surfaces gets faster and faster, and with increasing temperature, this process gets even more efficient, so we have a inverse temperature dependence in the water relaxation rate. And a good test to see this is making some plots. Here, a temperature dependent plot of R1 at some selected frequencies. And that what we see is that we obtain some apparent negative activation energies. So this is first time such plots are introduced by Jean-Pierre Korb. And so these negative activation energies can be explained with the, here the delta A is the AM minus the AS, and here the AM is the activation energy for translational diffusion, but we have here also the ES, which is higher than the first, and that is the potential binding energy to the surface. So again, if we come back to our representative sketch and think about the different possibilities, so actually for the water that is diffusing on the surface of the particle, here as you can see, since the Inulin matrix has many hydroxyl groups, the water during its translational motion on the surface can perform subsequent absorptions and desorptions, so it makes a fast chemical exchange. But as compared to the case on the particle surface, inside the pores, the water diffusion is much more slower. If you remember that we obtained 1 order of magnitude slower diffusion with respect to the water diffusion on the particle surface. So it is much more probable for the water inside the pores to perform, to involve in the chemical exchange procedure. So in this case, we can propose a relaxation model that we, in which that we particularly assume that the water inside the pores gets an efficient chemical exchange because they are diffusing much more slowly. And here, in the third term, we can write this contribution as here, where the P pore is the probability of the water molecule can exchange with the surface, and in the exchange dominated case, here the tau x, which is the chemical exchange time, is much more slower than the T1 of the water inside the pores. So, and before showing the fits, let me also very briefly mention about the reorientation mediated by translational displacement, that is the RNTD process. RNTD process is first time introduced by Kimmich and Weber, and it actually describes some sort of random walk of molecules on the surface of your material, and in this case, the molecule adapts its orientation according to the topology of the surface, and it involves into many absorption and desorption cycle, and it changes its reorientation, but at the same time it maintains its translational displacement, It can be, this RNTD process can be see as a two-dimensional diffusion that is limited on the gel surface in our case. So, and actually the spectral density function is given as the Lorentzian type contribution of all diffusion modes. That is weighted with some orientation structural factor. Here, this structure factor very much depends on the properties of the surface. And of course its geometry, and for the assumption of Q is equal to a constant, that is that all diffusion modes are equally weighted, we are reaching an equation shown here. And here actually we have two very important parameters. That is the high cutoff frequency and low cutoff frequency. So, and these frequencies are putting some limit for the water that can make its minimum and maximum molecular displacement. So for the high cutoff frequency, we can say that it is equal to the diffusion coefficients divided by the distance of closest approach. So in the best case, this is basically the radius of your, the diameter of your molecule. And for the low cutoff frequency, it is equal to the diffusion coefficient divided by the maximum molecular displacement. So if you have a confinement that one expects that this maximum molecular displacement corresponds to your length, confinement length scale. So in our case, that we can expect that this corresponds to the pore size, pore diameter in our gels. So again, if we come back to our relaxation model, so for the bulk water, basically we can assume that it is constant because it will not show any dispersion in the frequency range. And for the water molecules that is diffusing on close to the surface, we say that it involves into chemical exchange. However, for the simplicity, we can assume that it is given by some isotropic diffusion. Here, as you can see, it can be done by Huang-Fried model. And here in the Huang-Fried model, we, in order to reduce the number of free parameters, we here for the number of protons, we put the bulk water proton density, and for the distance of closest approach, we assume that it equals to 3.1 angstrom, and for the tau translational correlation time, here again for the simplicity, we substituted the diffusion coefficients that we have obtained from diffusometry measurements. And if you remember, we have said that the D2 coefficients that we can associate with the water on the surface. So we put these values into here and make this contribution also parameter free, let's say. And for this last term, as we have just mentioned, we assume that we have RNTD process. So, and here you can see the fits, and as you can see that even the number of water molecules that is diffusing inside the pore is small. Since the field cyclic relaxometry is very sensitive to the confinement, we see that in the decomposition of the individual contributions, that the majority, the biggest contributions comes from the waters that is diffusing inside the pores. And then we have the surface water, and then we have the bulk water contribution. And if we look at the output the parameters, here the most important are the high cutoff and low cutoff frequencies. The low cutoff frequencies is simply the frequency of where the spectral density, this R1, is make a breaking point here, and actually it corresponds to this expression. And if here we substitute the diffusion coefficients that we have obtained from diffusometry measurements, again, I remind you that we have associated the third diffusion process with the water that is diffusing inside the pore. So if we substitute these values into the values obtained from the feed, that we obtain a maximum molecular displacement corresponding to 8 to 9 nanometers, which is very close to the values that we obtained. The pore size we obtained with the bed measurements. And so in this case, one can say that this or fits also give information, some structural information about the gels that we have studied. Of course, one can run this in another way around, and so one can introduce the pore size as a fixed parameter, and look how the diffusion coefficients are changing, and one can compare it with the values that is obtained from diffusometry experiments. So before concluding my speech, so I want to show you that we also studied some different systems, for example, dextran microparticles, and mainly the similar scenario. We have some different fractions of water, and we have 3 different diffusion processes. And for example, here we have the inverse temperature dependence that is manifesting itself only at very low frequencies, and for the rest we have a normal behavior. In another example, we studied sucrose, and here we have totally expected behavior. That here we see that the trend of the relaxation water proton relaxation rate is normal, so that the chemical exchange is not so, let's say, efficient, but still we have 3 diffusion processes. And for the carboxymethyl cellulose, here we again have a normal behavior, except at the very low frequency range. And actually one can try different things. As you can see here, one can introduce different amounts of water, and one can in this way study the case of the that the pores that is fully saturated with water, or the partially saturated with water. So, as the conclusion, Inulin-based hydrogel microparticles were synthesized by microemulsion method using divinyl sulfone as crosslinker. The SEM images showed that the particles are in spherical shape and have a size distribution between 1 micrometer to 50 micrometer. From bed surface area measurements, the pore sizes of Inulin microparticles were found to have multimodal distribution. From thermal characterization, we see that the system show a glass transition at 85 Celsius and the degradation temperature at around 250 Celsius. From the high resolution NMR spectra and diffusometry experiments, the presence of different fractions of water is revealed, with diffusion processes much slower than the bulk water. And FFC relaxometry experiments show that the proton relaxation rate is around 20 times higher than the pure water at low frequencies, and it is dominated by chemical exchange. And we have fitted the nmrd profiles with a proposed relaxation model which takes into account both 3- and 2-dimensional diffusion for different water productions and chemical exchange at the water gel interface. This is the list of people that I want to thank you. I want to thank you to Professor Nurettin Şahiner in Çanakkale 18 Mart University and University of South Florida for providing me these samples. I thank to Professor Halil Mesut Öztop in Middle East Technical University in Ankara, Turkey. I thank to Professor Krug, Professor Pedro Sebastião in Lisbon, Maria Beira and Anand Kumar in University of Lisbon as well. And I want to thank you, Adam Rakowski in Institute of Molecular Physics in Polish Academy of Science. Thank you very much for your attention.
And we thank you for your lecture. And there's already a question there, and I notice with pleasure that the question has been posted by my colleague from Poland, from Krakow, actually, when I